All right. So we are on to order of draw for our Module 6 phlebotomy. I'm going to pop over to our Google real quick. All right. So we're going to have our lecture video. We're going to have some common test cheat sheet. I'm going to show you guys. That'll help you out when you get out and working. Study guide, the 2 slides, because this video will include 2 sets of slides, and then our Quizlet flashcards and our quiz. Okay. So first of all, what is order of draw? Order of draw in phlebotomy is the order of which your tubes should be drawn from the vein. Okay. So we stick the vein. We're putting on our first tube. It's telling you what tube should come first, second, third, fourth, if you have that many. Okay. So this is telling you exactly how you're supposed to draw them. Those tubes that you're using are determined by the tests ordered. Each test requires a specific tube. Sometimes one tube can be used for multiple tests. Once you know what tubes you have, then you put them in order so you know what order to use them in. You will memorize this order as you use it, okay? Each tube has a different additive inside of it. So not only does it have a vacuum built into that vacuutainer, it's got chemicals that react differently to the blood once it's in there. So the different types of additives equal the different types of lids or caps. You want to make sure you follow the proper order of draw to avoid the chemicals reacting differently in the blood and causing the results to be erroneous or wrong. So the very first one that you're going to draw always is going to be your culture tubes, okay? So on the list that you get, this one is straight from CLSI. Well, it's not straight from CLSI. This one's from the NCLEX, but I did compare it to CLSI, which is the Clinical Laboratory something institution. So the first one you're going to draw is going to be your blood cultures, lavender tube, serum separators. These guys are kind of all in the same category here, okay? So serum separators, red tubes or orange tubes, gold tubes, tiger tubes. All of those are going to be drawn next. Doesn't matter what order they're. Next, we're going to do our heparin tubes, which are the green ones. Lavender or EDTA tubes. If you have a pearl PPT separator, that one comes next. And then our gray fluoride tubes. So that's your order. Most order of draw lists or sheets you find, like Quest and LabCorp, all have their own too. It's going to start at the top with the first one you draw and then go down the list. Okay, that's how you read it. I will provide you, if you haven't already gotten it from me, with an order of draw card to keep in your pocket when you go to work. That'll have this listed so you have it there handy until you do have those things memorized. The next one is a blood tubes order of draw video. Ignore the order if it's different than this chart, okay, or the one I give you. This video just gives you more information about how the tubes and the additives react differently. It's good for information, but if the order is different than here, don't follow it. So your order of draw, the blood cultures are always drawn first, as we discussed. The cultures must reflect the natural state of the blood and composition. So if any other chemicals get in there, it does contaminate it and it'll cause it not to be accurate. Non-additive tubes, like the plain red or white ones, don't have any additives in it. Sometimes this may require coagulation, serum studies, and would result in cross-contamination if they're done after a blue one or, say, a lavender one. These are mixed up some. Okay, so no additive tubes come after blood culture, and then you have your light blue tubes. These are your sodium citrate tubes, usually for anticoagulation studies like PTINRs or INRs. The next one would be the rest of your red, gold, serum separators, all of those that have the additives with gel or without gel. Whatever the case is, if it's a serum separator with the silicone coating and the additive there, those go next. Green is the heparin, usually used for chemistry studies. Lavender would then come next, which is your EDTAs. Those are for complete blood counts and all of that stuff. That the lavender is actually kind of an anticoagulant versus a coagulant. These lavender tubes are also being replaced by pink tubes here pretty soon. The gray or potassium or sodium fluoride tubes are usually for glucose testing, and those are last. All right, so we have tubes with anticoagulants, so it stops the blood from clotting inside the tube. Those are your lavenders, EDTAs, pink EDTAs, the blue EDTAs, whatever EDTA anticoagulant. Gray tube, light green with heparin, our sodium heparin, dark green, and our yellow top ACD tubes. Order of draw combos. So the time values of coagulation tests may be altered if a green specimen or heparin is drawn before the light blue. That's why you want to make sure your light blue comes first. The heparin tube will also draw for electrolytes. And because an EDTA contains salt, it can throw off the electrolyte balance. That's why you have to do the green one before the purple one. The gray oxalate fluoride tubes should be drawn after to the EDTA because EDTA preserves the blood, and the oxalate fluoride does not. Lavender tube binds with calcium. So the EDTA tube binds with the calcium, and if it's drawn before a tiger, the calcium levels in the tiger tube can be incorrect. Okay. Make it show lower than accurate. The blood might not clot also because of the EDTA, and the SSTs, tigers or gel tubes require clotting. Heparin specimens drawn after the oxalate may cause cells to rupture and release potassium in plasma, causing potassium levels to be off. And exposed to glass, blood will clot. So sodium citrate shields the blood from the glass and prevents it from clotting. So when you get the blood into the tubes with these additives, you have to mix it gently. So inverting your tubes is important to making sure it mixes properly and it gets into all the blood evenly. So inversions means, okay, you have the tube upright, you turn it over upside down, and you turn it back upright. That is one full inversion. So it's up, down, up. That's 1, up, down, up, 2, up, down, up, 3. All right, so we do have some key terms, but before I go on to those, I want to go into our additional information about some of these tubes, okay? There's some info missing in the other slides, so I like to do this additional information for you guys just to give you a better idea of what's going on. We talked about the additives. Some are clot activators, some are anticoagulants. Tubes with clot activators will produce a serum sample when the blood is separated by centrifuge, which the centrifuging process is in the next mod, in mod 7. Tubes with an anticoagulant will produce a plasma sample after centrifuge. So before centrifuge, serum. After centrifuge, plasma, when there's gel involved. Some tests require the use of serum and some require plasma. All right, so blood cultures, we talked about those needing to be done first. This is a blood culture tube. It's got the long neck, so it pops into the hubs and is punctured by that, the rubber-covered needle pretty easily. There are 2 types of bottles drawn for every blood culture. You have anaerobic bottles for organisms without air, and then you have aerobic bottles for organisms with air. Okay, so how do we remember this? So an is a term or a prefix for not. So not air growth. So anaerobic, not for air. Aerobic is with air. Okay. Anaerobic, not air. Aerobic, air. So organisms that grow without air are anaerobic. Organisms that grow in air, aerobic. Inside each of these bottles are food. It's a medium. So the food in there or medium inside the bottle is for the specimens to be drawn into, and it feeds the bacteria so they grow. So if you draw the wrong thing, or you draw two anaerobics and two aerobics, you're not going to get the right growth in the bottles for them to be examined. When you're labeling your blood cultures, you want to make sure that the labels and the barcodes are properly seen, okay, and that they are labeled for each patient. You can't just label one, you got to label them both, and make sure that they are clear. So light blue top tubes, these are our sodium citrate tubes. They have a little liquid in there. That's the sodium citrate additive. It prevents clotting by binding with calcium. And if a green one is drawn before the blue, then it will alter the results. These tubes are anticoagulants and offer whole blood and plasma testing. The red, gold, and tiger tubes contain a clot activator and the gel for separating serum from the cells. All three of these tubes here are gel tubes. They're different sizes and colors, but they are all the same thing. They're all three SST gel with clot activators. The gel separates the serums from the cells for different chemistry, serologies, and immunology. And these gel tubes require centrifuging. They must be centrifuged within 8 hours. Different red, gold, or tigers with the centrifuge process. Sometimes it may be off due to different issues. So there's two specimens. The left picture here was placed into a horizontal position on the counter. So it's just laid down and left for a few minutes. So it clotted like this. So if you're thinking about the way it was laid down, let me see if I can turn it. Someone laid it on a counter like this, and the blood ran over and clotted in a horizontal fashion instead of it standing upright. As seen here, this one stood up and it started clotting, and it is now separated the way it's supposed to. Okay. You're supposed to set them up to clot before centrifuging. This is the actual centrifuge. There's multiple different types of centrifuges. The tubes can be centrifuged, and this will provide you with different specimens. Making sure that the centrifuge is done within that 8 hours is important. Once the centrifuge process is done, it's spun down. The gel actually comes from the bottom up to the middle above the blood cells, separating the serum and those blood cells. It's basically acting as a gel barrier. If the serum is a pinkish color, this could actually indicate damage to the blood cells or hemolysis. So serum separates from the red and white cells and collects at the top of the tube when centrifuged. Plasma separates when it's upon centrifugation. Oh, the same says the same thing. Actually, it's the same exact thing. Sorry, guys. So one comes before the other depending on the centrifuge process. Here, clot activators. So serum comes before centrifuging with gel. Plasma comes after. I apologize. I don't know why I got myself confused with that. There's more information in here. I'm not reading through all of this for you guys. This shows different levels of centrifuging based on improper speed. So if the centrifuge is not spinning fast enough, it won't even move the gel. So I'm going to look at these tubes from left to right. So the first one here, the gel is still down here at the bottom. So it didn't spin fast enough to move the gel at all. The next one, the gel did move, but if you see how it's still a little foggy here and there's still some cells on the top, it didn't spin fast enough to fully separate the cells out of the plasma into the bottom, or spin fast enough to fully get the gel separated. The same for the next one, but it was a little faster. There's still some gel here, but it is starting to weasel its way up. While this one, the last one on the right, was correctly centrifuged. You don't see any residual gel in the bottom section where the cells are, and we don't see any red left over in the top either. These are the opposite. So not the opposite, but from right to left, increasingly higher centrifuging G-force. This up here indicates some hemolysis or damage to the blood cells. That's why this is pinkish, but it's also not completely separated. So if you do see that pinkish color, just know that means it wasn't centrifuged, or that there's damage. Sorry. Red non-gel tubes are sometimes non-additives, so you want to read the label and see if there's an additive. If this tube is drawn after containing one with an additive, it can change the values. That's why if there's no additive, it should be drawn after the culture and before the blue tube. So that's where that came from. All right, so green, anticoagulation. If the green specimen is drawn before EDTA, remember the salts could throw off the reading for the electrolytes. The pink or lavender tubes are often used for whole blood specimens, and they do prevent clotting by binding with calcium. So whole blood specimens are for EDTA or lavenders. The gray top, sodium fluoride, most often used for glucose testing. And here's some more information if you want to go in and read that. I didn't read everything on these slides to you guys just because it's additional info. Come back and read them if you would like. But I am going to jump over to the common test tube cheat sheet that's linked into your mod for Module 6. The top here is the same as the bottom. So you got two on the same page. And what it is it's listing frequently done tests with the tube that goes with it. So frequently done tests, the tube that it goes with, test tube. If you print this out, you can actually keep it with you when you go to work, or make a screenshot, put it in your phone, whatever you want to do. This cheat sheet is here as a reference for you to make it easier when you first start. In module, I think 11 or 12, you're going to look up the tests and find the tubes as an activity. But for now, this is just a cheat sheet for you to be able to print. Just a reminder what some of those tubes look like. That's all this is, and reminders for inversions. I just thought this was a good example of the multiple different types of tubes you may see: rubber tops, plastic tops, different colors, different labels, cultures versus the other tubes next to it, all of that. So just check that out when you have time. And then remember, this is your order of draw. All right, thanks, guys. Go into your flashcards, study up on your tubes. I'll talk to y'all later.